Hantavirus, the full picture, Andes virus, the MV Hondius outbreak, and the vaccine question. What is hantavirus? Hantaviruses are a family of RNA viruses carried primarily by rodents, mice, rats, voles, which shed the virus in their urine, feces, and saliva without becoming ill themselves. Humans are typically infected by inhaling aerosolized particles from rodent droppings. The disease manifests in two main forms: hemorrhagic fever with renal syndrome, HFRS, predominant in the Old World, hantavirus pulmonary syndrome, HPS, predominant in the Americas, with mortality rates of 30 to 40% in severe cases. There is no specific antiviral treatment. Patients receive supportive care and hope their immune system prevails. The Andes virus, the exceptional strain. The strain currently making headlines is Andes virus, ANDV, and it is genuinely exceptional in the worst possible way. It is the only known hantavirus capable of human-to-human transmission. Every other strain requires direct contact with infected rodents or their excretions. Andes can jump directly between people through close respiratory contact, placing it in an entirely different epidemiological league from its cousins. The molecular biology of why only Andes can do this, likely related to specific adaptations in its nucleoprotein and glycoproteins, is still not fully resolved, and that question alone warrants a dedicated research program. The MV Hondius outbreak. Breaking now. This is a live and developing situation as of this week. The Dutch cruise ship MV Hondius departed Ushuaia, Argentina on the 1st of April, 2026, visiting Antarctica and remote South Atlantic islands. As of the 7th of May, 2026, 5 confirmed cases, 8 suspected cases, 3 deaths, 2 confirmed. Andes virus, key case timeline. Case details: Case 1, 70-year-old Dutch man, symptoms the 6th of April, died the 11th of April on board. Case 2, his 69-year-old wife, disembarked St. Helena the 24th of April, died Johannesburg the 26th of April. Case 3, adult male, evacuated to South Africa, currently in ICU, PCR confirmed. Case 4, adult female, died the 2nd of May, 2026. Case 2 is the critical epidemiological signal. The wife almost certainly acquired her infection through close personal contact with her husband, not through any rodent exposure in Antarctica. This is human-to-human transmission in action, and it has alarmed researchers. The MV Hondius has since sailed for Tenerife, Canary Islands, after Spain's Ministry of Health approved docking, despite objections from the Canary Islands president over safety concerns. WHO currently rates global risk as low, because human-to-human transmission requires close proximity and only limited spread among close contacts has been observed historically. However, the situation is being watched very carefully. A cruise ship carrying international passengers and leaving a trail of deaths across 3 continents is precisely the kind of event that focuses minds. Could an mRNA vaccine be developed? The short answer is yes, and the groundwork is already being laid, though the path is more complex than it was for COVID-19. Why the mRNA platform suits this virus? The mRNA vaccine platform is well suited to RNA viruses. The key surface proteins for hantaviruses are the GN and GC glycoproteins on the viral envelope, which mediate cell entry. You encode the mRNA sequence for these, wrap it in lipid nanoparticles, and let the body's own ribosomes produce the protein. The immune system mounts a response, memory cells form, and you have protection ready. COVID-19 proved this can go from sequence to clinical jab in under a year when urgency and funding align. The biological challenges: several factors complicate vaccine development for Andes specifically. 1. Immune evasion. Hantaviruses are remarkably good at suppressing early innate immune responses. They interfere with interferon signaling, buying time to replicate before the adaptive immune system mobilizes. A vaccine must prime a very robust T-cell and antibody response to overcome this. 2. Conformational complexity of the target protein. The GC glycoprotein undergoes significant conformational changes during cell entry, meaning the shape it presents to antibodies in a vaccine may differ from the shape it presents during actual infection. Getting the correct conformation into the mRNA construct matters enormously. 3. BSL-4 containment requirements. Unlike COVID-19, where hamsters and ferrets provided rapid feedback in standard labs, Andes virus research requires biosafety level 4 containment, the highest level, because of its lethality and transmissibility. This significantly slows experimental timelines. 4. Historically weak market incentives. Hantavirus outbreaks have been sporadic and geographically scattered. Pharmaceutical companies have lacked strong commercial motivation to invest, until now. Current research status: this is more advanced than most people realize. NIH's Rocky Mountain Laboratories, a BSL-4 facility, has been developing DNA and subunit vaccines against Andes and Sin-Nomba viruses for over a decade, with promising animal model results. Moderna and other mRNA platform holders have the technical capability to design an Andes mRNA vaccine rapidly. The GNGC sequences are well characterized. A 2022 study demonstrated that a single-dose mRNA vaccine targeting the Andes GN glycoprotein provided complete protection in Syrian hamster models, the best available animal model for HPS. That is a genuinely significant result. Chile and Argentina, where Andes is endemic, have been pushing for accelerated development. This outbreak will intensify that pressure considerably. Realistic timeline: with a COVID-19 scale emergency declaration and funding commitment, Phase 1 safety trial could begin within 12 to 18 months. Full regulatory approval, realistically 3 to 5 years. Given the need for efficacy trials in endemic regions, the BSL-4 constraint means challenge trials are not straightforward. Researchers would need to rely on natural exposure events in vaccinated versus unvaccinated populations, which is slower than a controlled trial. The mRNA platform dramatically accelerates the early stages. Sequence design and manufacturing scale-up are now nearly routine. The bottleneck is the clinical trial pipeline, not the underlying science. Also notable: the Andes virus made U.S. headlines in 2025 when an autopsy confirmed that Betsy Orokowa, wife of actor Gene Hackman, died of hantavirus, though that was a separate, rodent exposure case unrelated to the current outbreak. The bottom line: what makes this outbreak so scientifically significant is that Andes virus sits at a uniquely dangerous intersection: high lethality, human-to-human transmissibility, and no approved treatment or vaccine. The mRNA toolkit exists. The target proteins are known and sequenced. What has been missing is urgency, and a cruise ship outbreak with international passengers may finally provide it. Sources: WHO Disease Outbreak News. Don 599, https://